starting now with a new topic in our series. We're going to talk about neurons, neuroanatomy, neuronal functioning, and then we're going to talk about neural networks or connectionist systems that model, operate according to principles that are similar to the way in which neurons appear to operate. So we'll start by just looking at the basic anatomy of typical neurons. We'll talk about dendrites, soma or cell body, the axon hillock and the axon, the presynaptic terminals, glial and Schwann cells, terminal fibers, and synapses. We'll also talk a little bit about neurotransmitters, though not that much. After we get done with that, later in the lecture and other modules, we'll talk about some basic functional anatomy of the neuron, basic states of the neuron, basic function of the neuron, and basic types of neurons. So, when we think about a typical neuron, they have four morphologically or structurally distinct cellular components. On the one hand, they have a cell body or a soma, and that contains the nucleus as well as other structures related to their metabolic functioning. The soma also functions to integrate input from other neurons. Cell bodies have two kinds of processes, vaginations, extrusions. On the one hand, they've got dendrites. Dendrites are branch-like protrusions from the cell which reach out to other neurons to facilitate communication. They're, in essence, the input. Dendrites are shorter than axons, and they taper as they extend from the cell body. Dendrites also often exhibit multiple arborizations or branches. The axon is the extended protrusion from the cell body which acts to carry electrical signals from the cell body to other neurons. At the juncture of the cell body is the axon. The cell body and the axon is the axon hillock. That's the region of the cell body where the axon emerges, and the axon hillock, as we'll see, plays a crucial role. It acts like a gate to the axon and a trigger point for the primary functional role of neurons, action potentials. Now, in addition to neurons, our brains are composed of other cells. And the most important thing that we want to look at are glial cells. Glial cells break into two general groups, Schwann cells and oligodendrocytes. They wrap themselves around axons so as to create a sheath which facilitates the transmission of nerve impulses, and that sheath is called myelin. They also provide structural support for neurons. And in the mammalian brain, glial cells, or neuroglia, are cells that really are crucial to mental functioning. They provide the superstructure that supports the nerve cells, which aren't actually mostly connected, physically anyway, and they develop and maintain neuronal homeostasis by providing glycogen for energy, repairing damaged neurons, clearing away dead neurons, and maintaining the ionic balance outside the neurons. They sometimes even facilitate information processing functions of neurons. So, for instance, when they wrap themselves around the axons of neurons, forming that myelin sheath, that is a strategy for increasing signal speed. They also break down neurotransmitters in the synaptic cleft, as we'll see, and they even release neurotransmitters and ATP. The axon ends in presynaptic terminals or terminal fibers, branches from the axon which travel to other neurons and end with the synapses on those other neurons. Of course, synapses are just points of communication between one neuron and another. Chemical synapses are the most common by far, and a chemical synapse is actually a gap between one neuron and another, where the first neuron releases chemical transmitters, neurotransmitters, that bond with receptor sites on the other neuron. And once these transmitters bond, they affect the next neuron's state. Now, your brain operates using a wide variety of neurotransmitters, and neurons tend to specialize in the kind of neurotransmitter that they are sensitive to, or that they transmit to the postsynaptic neurons at the other side. These chemicals include things like serotonin, which is probably the most famous, or dopamine, also very popular. GABA is another example of this. And these neurotransmitters are released by one neuron. They bind to receptor sites on the other neuron, and they then facilitate the communication between neurons. And of course, different neurotransmitters have different functions. So as a sort of point of review, let's look at this through a sort of basic neuron picture that you find pretty commonly now. Postsynaptic terminals are receptor sites on those dendrites. The dendrites themselves are the protrusions from the cell body that have usually multiple arborizations. The cell body itself, or the soma, is the home to all of the elements of a neuron that control its metabolic functioning. So all its organelles and its nucleus and so on. The point at which the axon leaves the cell body, right, the long protrusion there, is called the axon hillock. When axons are myelinated, glial cells wrap themselves around the outside of an axon, and they always leave these little nodes of Ranvier, gaps between those glial cells, between those areas of myelinated axon. When we get to the end of an axon, we have the axon terminal, and ultimately that ends in axon end bulbs, or sometimes they're called synaptic knobs.